begin, sir?
Yes, ma'am. Please begin.
A very blissful morning to one and all present over here. I, Tasneem Baldiwala, working as an assistant professor, School of Pharmacy, Parul University. It's been privileged for me to extend my greetings to respected Dr. M.N. Patel, sir, Provost, Parul University, Dr. Abhay C, Dean, Faculty of Pharmacy, Dr. G.S. Chakrabarti, Principal, Parul Institute of Pharmacy and Research, Dr. Lalit Lata Jha, Principal, School of Pharmacy, and Dr. Mahendra Kumar, sir, Associate Professor, Department of Pharmacology, Maharshi Arvind Institute of Pharmacy, and my all dear faculty members and students. On behalf of School of Pharmacy, Parul University, I welcome you all dignitaries to grace the online national level webinar on reverse pharmacological approaches and scientific validation of ethnomedicines. As we all know about the emerging trends of ethnomedicines nowadays, ethnomedicines have broadly referred to traditional medicine practice concerned with the cultural interpretation of health, diseases, and illness. So, therefore, to show the importance and to share the knowledge of ethnomedicines, today we have with us Dr. Mahendra Kumar Chaudhary, sir, Associate Professor, Department of Pharmacology, Maharshi Arvind Institute of Pharmacy. Sir has more than 5 years of teaching experience, as well as Sir has given his valuable contribution in the field of research. Till date, Sir has more than 20 publications in more than national and international journals. To his credits, Sir has received Best Poster Presentation Awards from International Conference of Society of Ethnopharmacology at Dhaka, Bangladesh. Sir has also received Oral Presentation Award at International Conference of Society of Ethnopharmacology in Thailand. Furthermore, till date, Sir has attended more than 50 workshops. Till now, Sir has received 40,000 rupees grant from the university for the title research, that is synthesis and characterization of nanoparticles by green chemistry for its cancer potential. Furthermore, Sir has also received 40,000 rupees grant from the university for the research project HPLC fingerprinting and method development validation of ethnomedicinal plants. So therefore, today Sir has taken up the task to brief us for the topic, that is reverse pharmacological approaches and scientific validation of ethnomedicines. I heartily welcome you, Sir, for the webinar.
Well, thank you, Ms. Taslim, for introducing me, and good morning to all of you, scholars, then the ladies and gentlemen. This honor and pleasure it is for me to present the topic reverse pharmacological approach and scientific validation of ethnomedicines and some of our research outcome and further research scope for you. This live webinar means a lot for me since I am very enthusiastic participant in all the same kind of webinar and conferences, and I am very honored by invitation to deliver a keynote lecture on the webinar in the wonderful company of you all delegates and the moderator from the College of Pharmacy, which is conducted by the Parul University, Vadodara. So the pleasure and honor is all mine. I always pledge to do something for the incredible benefit for this particular field of reverse pharmacology and ethnomedicinal validation. And definitely this help to the scholars and chemists and those who are involved in this research, definitely they will get the benefit. So since today is the very auspicious day of the Guru Purnima, I would like to start with the mantra Guruhu Brahma, Guruhu Vishnu, Guruhu Devo Maheshwara, Guru Sakshat Param Brahma, Tasma Shri Guru Nama, to all gurus. So I would like to start the topic, that is reverse pharmacological approach and scientific validation of ethnomedicine. So over the past 2 decades, traditional medicine now also we can term as the complementary as well as the complementary as well as the alternative medicine, or we can say the local medicines or folklore claim considering medicine. So these medicine nowadays considering the considerable attention, and the complementary or alternative medicine has opened important, and it has the comprehensive as well as integrative health care provision, and it can reform the health sector. And if we can say that nowadays approximately 80% of the world population depends on exclusively on the plant and their products for the healing and for the health outcomes. A wide array of modern drugs, included the international pharmacopoeia, have an origin in ethnopharmacology and traditional herbal medicine. Various plants and their extracts and their ingredients have the unique pharmacological activities which have been evaluated for the various kind of activity like anti-inflammatory, anti-diabetic, anti-cancer, vasodilatory, antibacterial, antiviral, anti-convulsant, sedative, antipyretic, etc. So the reverse pharmacology integrates the documentation which included the clinical experimentation hints, and further we can develop the drug candidature as well as the formulation to the systematic and preciously. We can say the reverse pharmacology have the salient feature of, or we can say that it has the particular feature of combination of knowledge which learned from the traditional or folk medicine and the modern technology to provide the better, safer leads. So the scope of this particular field will be understanding the mechanism of action at multiple level of biological organization, as well as to optimize the safety, efficacy and acceptability of the leads in the natural products based on relevant science. So we can say while the classical pharmacological method is time consuming and sometimes it is inefficient, and using current technology and genomics, proteomics and metabolomics, scientists now move to the opposite direction, that is using the conventional pharmacological research for the drug development. So this strategy referred as reverse pharmacology, and it has many advantages. The drug development process with reverse pharmacology approach is much faster, means it not taking much time, and it is more efficient. And if we compare with the classical approach, we can say that we can get the result in lesser time. So the reverse pharmacology can be we can explain it is consist of the three main domains, that is, one is the traditional knowledge, and another one is the experimental observation and clinical experiments. So there are the three different phases of reverse pharmacology is there. First one is the experimental phase. In that, the first one we have to do the experimental part, documentation and the robust clinical observation of biodynamic effects of the formulation which is used in the folk medicine or already developed drug formulation and their precursors by the meticulous record keeping. Then we can say the second phase is exploratory phase, that is exploratory studies for the tolerability, drug interaction, dose range finding in ambulant patients with the defined subsets of disease, and paraclinical studies using relevant in vitro as well as in vivo methods or models to evaluate the target activity. And again, third one, the experimental phase. In that third phase includes experimental studies, basic and clinical at the several level of biological organization to identify and validate reverse pharmacological correlates of safety and efficacy of drug candidates. So the traditional knowledge inspired reverse pharmacology relates to reserving the routine laboratory to clinic path of drug discovery pipeline to the clinic to laboratory. It is a completely opposite. Thus, the drug discovery, generally based on the reverse pharmacology, follows a path from clinics to laboratories, and opposite direction, which is applied for the conventional synthetic drug development. So it is like in the conventional system, there is a molecule, then we are going for the experimentation part, then we are applying for the treatment of the people. But in the reverse pharmacological approach, the medicines are already used that we can evaluate for the scientific validation, for the evaluation of the different parameters by using in vitro, in vivo methods. Then we are going for the molecular level. Then again, we are going for the testing on the population. Since safety of the materials has already been established from the traditional use, so this track record will help to develop in the pharmaceutical development for the safety, validation, and other pharmacodynamic studies. So these studies can be conducted in the parallel. When we are talking about the area of cancer, so if we are taking a particular time frame since 1940s to the 2007, sorry. So around 155 molecules and around 73% are non-synthetic and 47 are actually being either natural products or directly derived therefrom. So the scientific validation of these ethnomedicinal plants or medicine will help for establishment and well documentation for folklore claim to the traditional healers. So one of the study we have conducted in association with the ICMR, National Institute of Traditional Medicine, and they have gone through a particular survey that local healers and traditional healers from the Western Ghat, which is the one of the biodiversity hotspot of the India. Out of two hotspot, one is the Himalayan region, another one is the Western Ghat. So in the Western Ghat, they have conducted a survey, and out of that survey, they evaluated and they met with the various local healers and traditional healers. They are using some medicine. They are claiming that these medicines have some potential. So we have differentiated and we worked with them. And finally, we identified some plants for the particular anti-cancer activities. And we have worked on this particular title, that is Evaluation of Anti-Cancer Activities of Ethnomedicinal Plants from Western Ghat. So about that study, I would like to explain. In that study, these are the contents. When we are talking about the cancer, this is the second leading cause of death worldwide as per the WHO, and various cancer are very deadly. And when we are talking about the India, around 2.5 million cancer cases are registered and various million deaths are occurred every year. The drugs which are used to treat the cancer, they are very harmful for the body until they are very specific for the cancer cell. And when we are using these drugs for the long time treatment, this drug will be associated with the several side effects. The use of ethnomedicine, or when we are going for the ethnomedicinal practices, so these attracted the attention to improve the healthcare outcome, which has enhanced to cure the various disease. And from the Western Ghat, various ethnomedicinal plants are used by the codified as well as the non-codifying practices. So codified practices, that is already documented, well documented, but non-codifying practice is also there, that is not well documented, but people are using, particular groups or people or ethnic people are using those medicine to treat their ailments. So, and they are practicing with these drugs without any side effect, but they doesn't have any scientific validation. So this scientific validation is still a question for various activity like immunomodulator, anti-cancer, antiviral, cardioprotective, hepatoprotective, and sometimes to strengthening the broken bones. So various plant that has been derived or that has been recovered or they identified from the Western Ghat, various plant is there and their various activities are performed scientifically. Also, the validation also has been done. So we have gone through the literature review, some plants which have been used by the traditional healers. Out of those plants for the cancer treatment, we have identified 2 plants, and after review of literature of those plants, we have found that 2 plants, that is, one was the Acanthus coyni and second one was the Clerodendron inermi. These plants were used the treatment of cough, fever, piles, urinary calculi, scorpion sting, anemia, wound healing, anti-tumor agent, and many other elements in traditional practices. Also, these plants has been evaluated for the anti-malarial, antioxidant, hepatoprotective, anti-inflammatory, antimicrobial, antiviral, hematological, and antihypotensive, or sometimes they use for the antifungal activity also. These plants also selected for the study, in that the people found, the researcher found that these plant content phytoconstituents have like olenolic acid, ursolic acid, betulinic acid, caffeic acid, and tannic acid. These are present, and these are known for the anti-cancer potential. So why the need for the study was there? So ethnomedicinal plants are there. They are used by the non-codified practitioners in various, also in the various codified classical dosage form also. The selected ethnomedicinal plants were used for the treatment of cancer, and that did not possess any scientific validation with the standard drug used in the modern medicine. So there are several scientific approaches there to evaluate the anti-cancer potential by in vitro as well as in vivo methods, and these methods are very well known, acceptable, and evaluating the anti-cancer potential of ethnomedicinal plant for its traditional claim. So in view of above investigation, we found that there is a need to conduct the detailed scientific validation and through studies for anti-cancer potential of this plant. So our objective was to evaluate the anti-cancer potential of ethnomedicinal plant by in vitro assay method, as well as the safety and efficacy of ethnomedicinal plant through anti-cancer in vivo models. So these 2 plant species we have selected. One was the Acanthus coyni family Amaranthacea, and second one was Clerodendron inermi family Verbenaceae. As per the International Union for the Conservation of Natural, they have listed these both plants in the threatened species, and out of that, the Acanthus coyni plant was endemic too. So people using, but there is a no conservation. So these are the red listed plant. Further, in the materials and method, the collection and authentication was done, and collection was done from the Dandeli Anchi Tiger Reserve from the Uttar Kannada and Sulebhavi from the Belgavi region, and the herbarium was prepared and submitted to the Regional Medical Research Center, that is ICMR Center, and we have received the voucher specimen number. And this is the schematic representation of work: identification, collection, and authentication of plant, followed by extraction by cold maceration method in methanol and water at the ratio of 70:30. Further, this was taken for the phytochemical investigation, followed by the HPLC evaluation for the marker compounds. Then it was subjected for the in vitro anti-cancer activity. In that, we have evaluated the extract for the dry exclusion assay and MTT assay using various cell lines like lung, breast, colon, leukemia, fibrosarcoma, Burkitt's lymphoma. And in the in vivo anti-cancer activities, brine shrimp bioassay was performed, and in the animal studies, oral toxicity studies was performed using the OECD guideline number 423, and 2 animal model was performed, that is Dalton's ascitic lymphoma model, and second one was fibrosarcoma model. So extraction, after the extraction, the extract was concentrated and dried and stored at the 2 to 8 degrees centigrade by using these machineries and process. And the yield of the plant for the clerodendron inermi, the percent yield of the plant extract was 1.84%, and for the Acaranthus coyni was 14.9, and nature of extract was brown and hygroscopic for the clerodendron, and dark brown and viscous and sticky for Acaranthus coyni. Some preliminary phytochemical investigation of plant extract also done, and these different tests was performed to find out which kind of moieties are present. Also, the extract was evaluated for the chromatographic analysis by using the HPLC Schematic LC20AD model with the C18 column at the wavelength of 210 nanometer at the isocratic mode with the methanol and water at the 90:10 ratio, and the pH was 5 using the glacial acetic acid. The three marker compounds, that is betulinic acid, olenic acid and ursolic acid, was evaluated, and the retention time was obtained, which was further matched, and further we have found the same RF value was there in the extract, which confirmed that this plant have the same moieties, as well as the spectrum also we have previously evaluated that I have not mentioned in this slide. In vitro studies, the cell counting, the live cell counting was performed by using the dye exclusion assay method. In that, we can check how much cells are live and how much cells are dyed, and followed by we have performed the tetrazolium dye assay, that is MTT assay, by the standard protocol. The assay was performed on the cancer cell lines, that is Burkitt's lymphoma, Dowdy cells, Fibrosarcoma, HT1080, lung cancer cell lines, that is A549, breast cancer cells, MCF7, colon cancer cell lines, HT29, leukemia cancer cell line, that is HL60. We have obtained the result, that is IC50 value for both plant when we are comparing with the standard dose. We have received the IC50 value on Burkitt's lymphoma cells, Fibrosarcoma cells, breast cancer, MCF7, colon cancer, HT29 cells, lung cancer, A549 cells, leukemia, HL60 cell lines. So the finding was there, if we are looking at the IC50 value of the doxorubicin, it is showing at the very low dose where the cytotoxicity was occurred. And also when we are comparing with the crude extract, it is not showing at the very low dose, it is showing at the very high dose, but it inhibiting the cell proliferation. So this outcome is indicating that these plants have potential to inhibit the cell proliferation. Further, we are come to in vivo studies. In the in vivo studies, we have performed one of the very interesting assay, that is Bryan-Shrimp bioassay or Bryan-Shrimp lethality bioassay. In that we grown the Bryan shrimps eggs and let them hatch. After the 24 hours of hatching, we have exposed the Bryan shrimps to the different concentration of plant extracts, and we have checked at different level of concentration of the plant extract how much the toxicity or how much percent toxicity we have found. For the Acaranthus coyni, this is the pattern at different concentration level, the percent cytotoxicity is there, and the IC50 value was found 100 ppm, below 100 ppm. The same results we have found for the Clerodendron inermi also. The IC50 value was found below 100 ppm. So this result finding, we can say these plants below 100 ppm, they are killing the 50% of population. So these plants can be considered as they have the potential of cytotoxicity. Further, we have evaluated for the animal experimentation also. For that, we have obtained ethical clearance certificate from the Institutional Animal Ethical Committee that approval was taken for the conduct of study. Then the oral toxicity studies, acute oral toxicity studies was performed by using the OECD guideline number 423 at the dose of 2000 mg per kg, and up to the 14 days we have observed there was no toxicity or no mortality was found. So we have considered the 1/10 of that dose, that it means 2000 mg per kg is 1/10 is 200 mg per kg. We have considered as a low dose, and double of that, that is 400 mg per kg, considered as a high dose. Tumor induction, we performed two kind of models. One was the Dalton's ascitic lymphoma model. Second one was the fibrosarcoma, that is solid tumor model. So in the case of Dalton's ascitic lymphoma model, cell suspension of 0.2 ml of 2 into 10 to the power 5 cells per mouse was taken from the tumor bearing mouse and injected intraperitoneally to the animals. And after 5 days, animal will get the tumor, and that animal we can include for the study by the day 1 only. And in the case of fibrosarcoma, that is solid tumor model, this model is the chemically induced cancer, or we can say 3-methylcholanthrin at the dose of 200 mg induced model. So the 3-methylcholanthrin, the carcinogen, was introduced at the dorsal region by the subcutaneous route, and after 2 to 3 weeks of the injection, the tumor was found on the mouse. The tumor size was increased, and for the injection of the cells to the other mice, we have excised the tumor and minced in a particular buffer saline solution and make a cell suspension, and the same cell suspension at the same time we have injected by the subcutaneously at the dorsal region, and further to the more number of animals, because sometimes we have found that we are not getting the tumor in some mice. So when the number of mice according to our groupings, they develop the tumor, we are separating them. When the size of tumor is 0.5 centimeter, we are considering as for the model, they are fit for the model. So the group discussion for the cancer model was taken as per the, you can see on the screen. Total 7 groups were distributed, and each group contains 6 animals. In the group number 1, they have received the normal saline. Group 2nd was the control group. Group 3rd received a standard drug doxorubicin at the dose of 2.5 mg per kg. Group 4 and 5th was received acaranthus coine extract at low dose and high dose as 200 mg per kg and 400 mg per kg per oral. And group 6 and 7 received the clerodendron inermi extract at low and high as 200 and 400 mg kg per oral. When we are talking about the group 3rd, that is a standard drug doxorubicin. In the case of Dalton's ascitic lymphoma model, the intraperitoneal route was selected, and the first dose was given after the 24 hours of the tumor insertion, and later on every 4th day. So that study was total 15 days study. 10 days total. Every 4th day they have received the standard drug. In the case of fibrosarcoma model, IV route by the tail vein was selected, and first dose the same like ascitic lymphoma model we have given after 24 hours and on every 4th day. The study duration was total 15 days. And different parameters we have evaluated like body weight, tumor volume, RBC count, WBC count, percent hemoglobin count, Kaplan-Meier survival plot, median survival time, and increased lifespan. The statistical analysis was performed for the entity results by interpretation of or also transform the data and nonlinear regression analysis by using the graph pad prism. And for the experimental animal experiment, we have used the ANOVA followed by the Dunnett's multiple as the standard test. So when we are talking about the animal activity in the Dalton's ascitic lymphoma model in the Swiss mice. So the body weight parameter was evaluated. So the body weight was measured before the experiment and after the experiment. When we are comparing the control group with the normal group, there is a significant increase in the body weight was found. And when we are comparing the doxorubicin treated group with the control group, there is a significant decrease in the body weight was found. And in the case of Acaranthus coineus at low dose, there is a reduction in body weight when we are comparing with the control group, but that reduction was non-significant, and there was a significant decrease in the body weight for the rest of other groups like Acaranthus coineus at high dose and Clerodendron inermi at low dose as well as the high dose. Other parameters like RBC count, WBC count and percent hemoglobin count was performed. In the case of RBC count in the Dalton's ascitic lymphoma model, when we are comparing the control group with the normal group, what we have found, there is a reduction in the RBC count. But when we are talking about the WBC count, there is an increase in the WBC count when we are comparing with the normal group. And the same, further the percent hemoglobin also reduced when we are comparing with the control group. Now we come to the doxorubicin treated group. This group, when we are comparing in the all three parameters, we can see that there is a significant decrease was found when we are comparing the doxorubicin treated group with the normal as well as the control group. The same three parameters when we are talking about the Acaranthus coineus at the low dose and high dose as well as the Clerodendron inermi low dose and high dose, there is a significant decrease was found when we are comparing with the control group. This was the finding of the exposure of this extract on these three parameters. Now the tumor parameters, tumor weight and tumor volume was observed when we are comparing the doxorubicin treated group. In the case of tumor weight and tumor volume, there is a significant decrease was found. And when we are comparing the Acaranthus coineus treated group and we are comparing with the control group, so there is a decrease in the tumor weight and tumor volume, but that decrease in the weight was not significant. Rest of the groups, treated groups like Acaranthus coineus at higher dose, that is 400 mg per kg, and Clerodendron inermi at 200 mg per kg and 400 mg per kg groups, there also we can observe that there is a significant decrease in the tumor volume as well as the tumor weight. Also, we have evaluated for the Kaplan-Meier survival plot. This provides the survival proportions. We can get the when the animal death events were found, how many subjects were survived at different days. So complete profile we can obtain by using this plot. And when we are evaluating the percent increased lifespan, there is a increase in the lifespan for the doxorubicin treated group as well as the both plant like Acaranthus coineus and Clerodendron inermi was found. Now we should come to the next model, that is Fibrosarcoma, that is solid tumor model in the Swiss mice. So in this model also we have evaluated for the body weight before the experiment and the after the experiment. When we are comparing the normal group and the control group, there is a significant increase was found in control group and significant decrease was found in the doxorubicin treated group. Means that doxorubicin is reducing the body weight after the experiment very significantly. And when we are comparing with the plant extract treated group, there is also reduction was there, significant difference was there in the body weight. RBC, WBC and percent hemoglobin count. As we know that Fibrosarcoma model for the evaluated for the RBC count, WBC count and percent hemoglobin. When we are talking about the RBC count, the control group when compared with the normal group, there is a significant decrease in the RBC count was found, but not in the case of WBC. There is a significant increase in the control group when it compared with the normal group. Here you can see directly And when we are talking about the percent hemoglobin count, there is also significant decrease in hemoglobin content when we are comparing the control group and normal group. Now we are come to the doxorubicin treated group. In the all three parameters, there is a significant decrease was found when we are comparing with normal as well as the control group. And rest of the treatment group like Acanthus coini at 200 and 400 mg per kg, Clerodendron inermi at 200 and 400 mg per kg body weight, we have found that there is a decrease is there when we are comparing with the control group, but that decrease in the RBC, WBC and percent hemoglobin count, that was not significant, like if we compare with the doxorubicin. So in this slide, you can see the tumor size of the different groups like control, doxorubicin treated, Acanthus coini at higher dose and lower dose, and Clerodendron inermi at higher dose and lower dose. The tumor size was measured after the interval of 72 hours for the period of 2 weeks by using this particular formula. And after the tumor progression, this parameter was evaluated. So in the tumor parameters, the percent tumor burden was evaluated. When we are comparing the doxorubicin treated group in the case of tumor burden, there is a significant decrease was found. But when we are comparing with the Acanthus coini as well as Clerodendron inermi, there is a decrease in the burden, but not as the doxorubicin. This was the findings. This is the pattern of tumor progression, how the doxorubicin treated group, tumor containing or tumor bearing mouse, the tumor burden was there, tumor progression was there, and in the other groups, how the tumor progression was there. Also in the downside, the figure is there in that the excised fibrosarcoma tumors of the selected group are mentioned or denoted. So when we are talking about the very important survival plot, that is Kaplan-Meier survival plot, in that we can know the survival data summary. In that there is a increase in the median survival time. Easily we can see there is a median survival time is increased. And when we are talking about the percent increased lifespan, there is an increase in lifespan, doxorubicin as well as in the other treatment group. That is the finding. So summary of this particular research work is the selected ethnomedicinal plant extract have anti-cancer phytocompounds which exhibits the anti-proliferative potential that were evaluated by different parameters such as MTT assay using the different cancer cell lines, and the IC50 values, that value suggests the plant extract inhibit the cell proliferation. Although we cannot compare the plant extract with the standard drug doxorubicin, these extracts are not showing the same or the same efficacy like doxorubicin, but they are sensitizing the cell proliferation. So that is the base for the understanding which molecule is behind this particular activity. The in vivo study also we have performed, and by using the branching lethality bioassay, that result suggested both extract have the IC50 value below 100 ppm, and which shown that Acanthus coini and Clerodendron inermi, both plant extract have toxicity. Dalton's ascitic lymphoma and fibrosarcoma solid tumor models in Swiss mice are the very standard models, and these models shows the ethnomedicinal plant extract not destructing the hematological content like RBC, WBC and percent hemoglobin count as doxorubicin treated group. So when we are comparing this group with the control group, these are inhibiting the tumor progression as well as the increasing the lifespan of treated mice. So all the parameters from the suggested and selected ethnomedicinal plant extract, these extracts sensitize and possess the anti-cancer potential in dose-dependent order and that has been used as a folklore medicine that provides a scientific base for the validation to its traditional claims. So the outcome of the study, the findings suggest that the selected ethnomedicinal plants and their extract enriched with multiple biotherapeutic properties and surely to be utilized as emerging plant-based medicine in the form of phytopharmaceuticals in diverse verticals of cancers and related areas. Also, the present outcome suggests that the new and unexplored ethnomedicinal plant from the particular region, that is northern part of Western Ghat, could be the potential probable drug discovery for the preparation of scientific database. So if I should talk about the future scope also, for this particular study, the future scope is the lead molecule which are present in the plant might have the further scope of interest, which phytochemical characterization, diversified mechanistic approach for identification and specific leads, and the study can be in the direction to change the cellular integrity parameters and checkpoints using cell cycle analysis, that is flow cytometry, for the cause of apoptosis and various cellular cascade can be scope for a possible mechanism of the anti-cancer potential of ethnomedicinal plants. Formulation and development of dosage form where these plants were used, the quantification of active lead molecules also an area for the future work. So this work we have done, and out of that work still we have published 2 publication and other one also in the line. And these are the presentation which I have given related to this work, and some trainings also I have gone through, and some of thing I should say that some techniques are not available at very many institutes. So some techniques I have learned from specific institutes like Bhabha Atomic Research Center, TIFR, or specific instrumental techniques from the particular industries also. So these are the 2 publications and some certificates where I have gone through the training, and these are the work outcomes and some conferences outcomes where I have presented. So at the end, I should say that ethnomedicinal, the particular region is still unexplored region is there, and many folklore medicines are there which have potential to treat many diseases, but still they require the scientific validation to compete the modern medicine. So with this word, I would like to complete my session here only. Thank you.
Thank you, sir, for this brief and detailed introduction regarding ethnomedicines, its validation, and reverse pharmacological approaches. The session is open for questions. If any participants is having any query, the queries are most welcome. Okay, so thank you, sir. So on behalf of Parul University School of Pharmacy, I would like to felicitate sir with the EAT certificate. Sir, with your permission, I would like to share my screen.
Sure. It is a very honor for me to given this presentation in this webinar. And if we are continue we are in this field, so definitely some outcome will be there. So thank you to the moderator as well as Shehzad sir to conduct this very wonderful webinar. And I also would like to thank to the Parul University to they are conducting the such kind of good seminar to enrich the knowledge, or they can across the knowledge, they can disseminate the knowledge to the different domains and as well as for the research scholars. Thank you very much.
Thank you so much, sir. Sir, actually, I just wanted to know one thing, that in cancer you have used two models. Like, so is it necessary that we should have this comparison data, or through one model also it is sufficient data we can extrapolate?
Actually, the traditional healers, they are using the same drug for the different tumors. They don't know which kind of tumor is there, which kind of specialized cells are affected with the tumors. That's why we are trying to evaluate the same drug with the more models. One more model I have evaluated that I have not discussed in this session because of the time constraint. That one was the radiation induced thymic lymphoma model. So sometimes because of the radiation also people are getting tumors. So this was the reason, that's why I have evaluated this drug molecule for particular more than one model and more than one cell lines I have used. But if you are damn sure that the particular drug or particular entity is there that can target for a particular or a specific type of tumor, you can evaluate one model also.
Thank you so much, sir. I'll be just sharing the feedback link for the session. So the participants will be getting an e-certificate once they fill the feedback links.
Yes, definitely.
So the link for feedback link is shared in the chat box. So I request all the participants to kindly fill the feedback links. And it was again a wonderful session from sir. Again, if anyone would like to have any clearance for any topic or any word regarding the presentation, they can interact with sir. So I hope the session was informative for each and everyone. So we can conclude the session if anybody is having no queries. Thank you, sir, once again from School of Pharmacy for providing us knowledge regarding the ethnomedicines and reverse approaches in pharmacology. Thank you, one and all.
Thank you. Thank you.
Thank you.